So hello everybody, I hope you can hear me. I don't see anyone. So if you are nodding or whatsoever, I won't be able to know whether you can hear me or not. So it would be nice if some of you could switch your video on, so I'm not only talking to a wall. And you're seeing here a little quiz to sum up some of the last lectures I gave to you. And now I'm just looking myself because and if somebody of you got the result, oh, I'm already one too far. I don't know what happened here. You already see the result now. So I was clicking on something while I wanted to click on I was looking for the chat function. I can't see it. Why can I not see it? I want to find this now because if somebody, here it is. If somebody has a question or whatsoever during the seminar, during the lecture, such that I can keep an eye on it. So I hope all of you guys are doing well. So this little quiz that I already clicked the result on. The first question is, these species don't have sex. How are they called? They're called isogamous because the gametes do have the same size. So iso, same sized. Second, not all species have a fixed mating system, but there is variation. Third, genetically determined decision rule leading to a tactic. Tactic is the behavior we observed, and here the result would be strategy. And first, the species that lives in polytrinaandrous groups with multiple males, multiple females would have been the baboons. And so the result is if so. If so is the abbreviation for intraspecific variation in social organization, and it will be one main topic in the lecture today. So, oh, that is still wrong. Why is it still wrong? I thought I changed this. Last week we talked about, what was it? The care system. 2 weeks ago, the mating system. And today we're going to talk about social organization and social structure. Should have been already addressed to you by Cedric Suer earlier this semester. So this by Sosa, that is wrong. It was last year. And I will also quickly address it either today or during the lecture tomorrow. The point being that the social network analyses that are done by Cedric and his students are an important aspect of how to understand social structure with modern methods. But I still would like to give an overview. What are the different topics that are addressed there that might not be so easily evident in the social network analysis. So trying to continue here. This is some repetition about the lecture on mating system, and I do not know how other lecturers are doing this, but when I was getting trained about how to lecture and how to teach students, but also kids, as I also did some studies on how to become a teacher, it is clear from psychology, one of the main ways to learn is repetition, and thus I always try to put some repetition in my lectures. You will see that my lectures, if anything, they might rather have too much than too little information. So it's especially important to repeat the information that I think is most important. Also, I only found out, I think this weekend, that the PDF I had sent of my lecture from last week had not been uploaded on Moodle. And if something like this happens with me or with anyone else, then you have to raise your voice and say, I'm sorry, we don't have the PDF or the PowerPoint, because normally, at least in my case, when I do lecture, I expect that you have the slides in front of you and you can make some notes in there, because the way how one is lecturing, of course, differs a lot with whether you do have or do not have this information. But may I quickly ask, you have the PDF from this lecture now? Yes, no? I see Victor is nodding, so it should be on Moodle now. So I assume you have it. So how the sexes would maximize their fitness, it's about evolution. So for males, they would go around and copulate with many females that stay home and take care of the offspring, while the male themselves look for more females. For the females, it would be the other round. They mate and then leave their offspring, could be the eggs, the chicks, the pups, with the male who should take care of the offspring to increase their survival probability and their growth rate, while the females go foraging for more food to get more resources to be able to produce more eggs. And obviously, either both of this can happen at the same time, and this leads to conflict between the sexes. There are different evolutionary interests, and the outcome of these conflicts determines the mating system, but also the social organization and the care system. And today we talk about the social organization and what this is. Social organization is a composition of social units and the different tactics individuals follow to maximize reproductive success determines the composition of these social units. So social organization, that's group composition or the composition of social units. The group size, is it 20, 200 individuals? And also very important is kinship pattern. Are the individuals, if there's a group, closely related to each other, or are they strangers? So it's a difference whether you have a baboon group where there's different female matrilines and the males are all not related to each other, or whether you have, let's say, a meerkat group where you have one breeding male, one breeding female, and their adult offspring, and they're all very closely related in a family group. So this has, of course, strong consequences for the entire social system. So for social organization, we ask how many adult males and females are in a group or the social unit. We only consider adults because offspring is rather a component of the care system. And that could be solitary living. Solitary living, important here is to note, it must be both males and females that are solitary living, because in most species, if in nearly all species, you have solitary dispersing individuals. And this, if you have solitary dispersing individuals, it doesn't mean the social organization can be solitary living. It's just a specific life history phase. It can be pair living, one male, one female, breeding together with or without offspring. But as we heard 2 weeks ago, breeding together doesn't mean that there are no extra pair paternities, but in fact, extra pair paternities can be very high in many families of birds, but also in mammals, it's considerable. Then subgroups are families. So families would be that the offspring remain at home and family groups are forming. Then group living can be one male and multiple females, multiple males and one female, multi-male, multi-female groups. Or what's also possible, there's groups consisting only of males and groups only consisting of females. The two sexes are segregated. This we, for example, observe in a few species of ungulates, of even-toed. Even-toed. Now I missed the English word. Even-toed ungulates, that's correct, and they are called artiodactylus. It can be sex-specific. Sex-specific means the females might be living in groups while the males are solitary. We find this also again in many artiodactyla, for example, in the impala and some other gazelles and antelopes, where the females walk around in groups and the males are solitary living and defend territories through which the female groups are migrating. Could also be that the males are social and the females are solitary. Though I do not know of any. I mean, the cheetah would be one example. In the cheetah, the females are always solitary with the offspring maybe, but you find male coalitions. These are especially close related males, brothers, half-brothers, that stay together, 2, 3, 4 males, and that hunt together, and that also look for females together. So that would be one example where the sex-specific social organization could be females solitary and males living in groups. Of course, these forms of social organization are related to the mating system, and often they are confused because it kind of indicates what people think the mating system is. And so the question is, how is this influenced by the mating system, and how does this influence the mating system? If a male lives together with many females, it might be different than if he lives together with only one female or no female at all. And the other question is that's rising here is, why do animals live in groups at all, or why do they live solitarily? So the mating system of a species, as I said, can be related to the social organization. So solitary living species where males live alone, females live alone, they could show promiscuity or polygynandry. That means one male and females are mating with several females and females with several males. And if there's some mate choice, we call it promiscuity. So the female doesn't mate with any male, but only with the most attractive one. Whatever makes it attractive can differ from species to species. Now that would be polygynandry, sorry. While promiscuity would be that there's no mate choice, like we, for example, find in coral, some invertebrates, also in the snowshoe hare. When they're living in pairs, it can be monogamy or polygynandry. It can be that the male, for example, lives in a pair with a female, but he has a secondary female in a secondary territory. If it's one male, multiple females, it could be polygyny or monogamy. I mean, it doesn't mean when there are several females in the group that he mates with all of them. It can be that there's only one dominant breeding females and the other females cannot breed. Multiple males and one female could be polyandry or monogamy. It's the same like with the males. When there are multiple males and multiple females, at least theoretically, everything is possible. So it could be that there's only a pair mating. It could be that one male mates with several females. It could be that one female mates with several males, or that everybody mates with everyone. So from only observing this in nature as a social organization, from only seeing there's multiple males, multiple females in a group, you cannot make a real conclusion about what the mating system really is. For example, you might observe when we talk about primates, multi-male, multi-female groups. In baboons, they are polygynandrous. But you see the same also in callitrichid monkeys, like how they call it, Callitrix chachus, or Gerdes monkeys, common marmosets, they're called. There you also find multiple males, multiple females, but there's normally only one breeding pair, and the other ones are non-breeding hairpairs of both sexes. So if you see multi-males, multi-females, you know nothing else about the how they interact with each other from the species. You cannot make a conclusion about the mating system. One male and one female, so only male, only female groups could be promiscuity or polygynandry, and the same when there are sex-specific social organization. So, yeah. Okay, so how does the mating system relate to the social organization? This is about economics of female monopolization, which is influenced by female group size, female homage size, and seasonality of breeding. It means, like I said at the beginning, males are normally evolutionarily driven to try to mate with as many females as possible and not to let other males mate with them. And so they might have fitness benefits of defending groups of females, but whether or not it is possible to defend groups of females depends on whether are there groups and over which area do these groups range, how big are the groups, which will all be factors influencing whether for the male it makes sense to try to monopolize a group or to be part of a group with multiple other males, or to have maybe a solitary territory and mate for the female groups to walk through. So how does this influence the mating system? How does this influence the social organization? And why do females live in groups are important questions here. And this is what I always like to emphasize, also a question, why do females live solitarily? It has often been assumed, and I will come to this at the end of the lecture today, that solitary living is kind of the default stage. It doesn't need any explanation, but with increasing evidence from field studies and comparative studies, we find more and more indication that living solitarily might not be an easy solution. It raises other important problems animals have to cope with. And thus the question rises, why do animals sometimes live solitarily? So to explain all of this, what has been traditionally been used, and especially in the field of primatology, is the so-called socio-ecological model of social organization, the center of which is the distribution of females. And it is assumed the females make the best choice to have sufficient resources to raise offspring, and then the males follow the female choice, and depending on what the females do, they follow their specific tactic, and this then determines the population's form of social organization. So distribution of resources, that is especially food, but can also be shelter, can be places where it's safe to raise offspring, influences female distribution. If there's little food, then they might be spread out solitarily. If there's lots of food, then they might be able to form groups and benefit from group living. Predation risk is another important factor because living in groups can reduce predation risk, and I will show several examples just later. So with a high predation risk, and if group living can be a tactic against high predation risk, it favors the formation of groups. So this both the distribution of resources and predation risk determines the outcome of the net benefits of group living, the benefits minus the costs, and then influences female distribution. When the benefits are higher, then they are more likely to form groups. If the benefits are lower, then the costs, if there would be group living, then they are rather solitary living. So here we have many competition for resources. And if there's lots of food, then there will be little competition. If there's little food, there will be a lot of competition. And here, the advantage of not being alone in the case of a predatory attack. Then there is the risk of infanticide that influences it, because competition between females can lead to female infanticide. It would induce a risk, but it can also be that the risk of infanticide comes from the males, and living in groups can help the females to counter this risk. And then, like I said, finally, the distribution of the males, the distribution of the females determines the distribution of the males. And then there could be benefits of paternal care, but normally, and this is assumed now, the benefits of paternal care only come secondarily. If males do live in groups or in pairs, then the question rises, does it pay them out to, in addition, show paternal care or not? There's no good case where people would say males started forming pairs because of the benefits of paternal care, but this only comes secondary. If a male lives in a pair, and then he might have time and energy left to show paternal care to increase his own reproductive success. So what are costs of living in groups? So should not everybody live in groups? I mean, groups are more conspicuous. Groups is easier detected by predators, and this can lead to solitary foraging. For example, this guy here, the African striped mouse, it's a small mouse. And if you imagine they live in open habitats and the areas, and if you imagine a herd of mice running around, it would be very conspicuous. And then predation risk would increase, and this could be quite costly. Rodents that you see living in groups often have a colonial system where they never go far away from the burrow. So they might be able to warn each other, and maybe the predator sees, okay, there are 100, or let's say 20 marmots, or 20 whistling rats, or 20 pikas, but they are very close to shelter, and then they have the benefit of group living. If one of them detects the predator from above, he gives a signal, typically a whistle. Both the marmots do that, the whistling rats do that, pikas also do that, and then they can quickly disappear. But like most rodents, like these mice, they don't live close to burrow systems. They go and forage all over the area. They wouldn't have this benefit. Here's the striped mouse. There you see how it looks, and it lives in this open habitat, and a group of striped mice running around would be quite conspicuous. The only time when you see them in groups, like in this picture here, is in the morning when they're basking in the sun next to the nest. And next to the nest means it's like a colony of rodents. They can quickly disappear into the safety of the nest. So we have the same like a marmot colony or a peacock colony. In this case, when they are found in groups, the escape area is very close. So what they do, they benefit from group living. I will show you this later, but they do show solitary foraging. They go alone for to look for food, with this reducing predation risk and thus costs of group living during the day. Other costs of group living is, of course, competition for food. If there's not enough food, then and then you live in a group, then of course it induces costs. Competition for mates. So individuals compete with each other for the access to mates, and this can then induce costs. And I don't have to tell you this, you know it from your own experience, that I think many of you have now to be in quarantine for some time, the risk of transmission of disease. When you are alone, you have a low risk to get a disease because where should it come from? But when you live in social groups, this is where diseases can spread from individual to individual. This has been studied in the beginning of behavioral ecology in the 70s and 80s and later in many species, like here these prairie dogs. These are also rodents, quite related to the marmots. So these are ground living squirrels. And here it was found that there are more ectoparasites in large than in small colonies. So the larger the colonies, the more ectoparasites they had. Ectoparasites, these are fleas and lice and ticks, basically. In birds, the same. Here in these swallows, swallow bugs per nestling. And these bugs, these are again, they're called bugs, but they're ticks, I believe. They are arachnids, not insects, but now I'm not sure. I think there are some ticks, like some arachnids. And here you see the colony size from one means not one individual, but it means one pair, so 2 individuals, up to more than, what do we have here, around 3,000 nests, so 3,000 pairs. And here's the increase of these parasites, not this time, not like in the prairie dogs counted in the fur, but in the nest of these animals. And in the nest, this is where they get access to the chicks and they drink blood from the chicks. And this is, of course, very, very costly, and it can have significant effects on the growth, and with this later survival probability and reproductive success of the chicks of a pair. Other possible costs when you live in a group is egg dumping or intraspecific brood parasitism. If you live alone and you have a nest, and then you know what's going on in your nest. But when you live in a group with 10 other females and you lay a couple of eggs, it can be another female dumps an egg into your nest, and you will raise the chick that's not yours and invest in it with parental care, even though it's not yours. And animals, typically, they cannot count to numbers higher than 3. Of course, there are differences from species to species, but a female that has several nests in its nest will not be able to realize it when instead of 3 eggs, there are 4 eggs. And even if it could be, it couldn't differentiate of the 4 eggs, which one is its own and which one not. So even then it would be too costly to abandon the entire nest, because there's 1 egg that's not hers and she would have to invest in all the offspring. And species, this is quite common in different kinds of bird species, this intraspecific egg dumping, for example, in wood ducks, canvasbacks, redheads, common eiders. These are also ducks. I should have put this in a better order. Tree sparrows are also starlings. You might know starlings. They also like to, I have some starling nest boxes in the garden because they like to brood in colonies where many pairs live together. And also there is the risk that one female puts its egg into the nest of another female. And with this gives the other female or the other pair even the costs of parental care. Acorn woodpecker, there was just a very nice paper about them in Current Biology last week. This is an amazing study of cooperatively breeding birds from the United States. These woodpeckers, they are storing food. These acorns, between little holes in old dead tree trunks, and these acorn wood banks can have thousands of these acorns in it, providing food in the season of low food availability to both the adults and the offspring. And there is quite some risk of females not egg dumping, but destroying each other's eggs. These are typical. There might be several females breeding together. They are typically closely related, and more than one female breeds, and they all take care of the offspring, including a larger group of males. So they all breed together, and the female that lays the first clutch of eggs has a high risk that other females, even though they're close relatives, go into the nest and destroy her egg, like you see here in this picture. This is called egg tossing. And the paper that came out last week or 2 weeks ago in Current Biology is about these acorn woodpeckers. And there is strong competition for breeding. There's only a few females per group that are allowed to breed. And sometimes it's only one female. And when a female dies, then there's competition. Who takes over the breeding position? And the breeding position is taken over by females from neighboring groups. And then you have coalitions. That means you might have the group in the center from your observation, and here the female dies. And then females from neighboring groups come and fight with each other for these breeding positions. And this can happen over 2 to 3 weeks. So you might have from one group, there are 3 females coming, and from the other group also 3, from the other one 4 females, 2 females, and they fight with each other over 2 weeks until one coalition is winning and taking over the breeding position. And more interestingly, while they're fighting, there might be females from other groups also coming, not participating in the fight, but observing what's going on to get some social information about what is happening there. So you have these fighting females, which the authors called warriors for whatever reason, and the spectators. And for the spectators, it's very interesting to observe these fights of neighboring groups. And the assumption here is when you observe, let's say, 3 groups fighting with each other, and you see who wins, you also see who loses. And when you see who loses, it might be a signal for you that you, with your coalition partners from other group, you might rather go and try to attack this group and get the females out of there to take over the breeding position than other groups. So this is some kind of social organization. And so what I've been talking about now for the last few minutes would thus be not a component of social organization, but of social structure, how individuals are interacting with each other. So these are the costs of group living. So that was the last one, risk of infanticide by group members. That is also quite common in mammals, for example, in house mice, also wild living house mice, and they often females kill each other pups. We observe this in striped mice. It's also especially the females, or here in these banded mongooses from East and South Africa. These are all all communal breeders. Communal breeders means there are several breeding females that help each other in raising the offspring. But in all these species, the female that first gives birth has a high risk that other females kill her pups to increase the investment of the group into her own pups, relatively seen. And this we studied in the African striped mouse, my main study species. And for this, we studied them in the field, but we cannot really see in the field what's happening in the nest. So we had here a captive colony that's at the research station in South Africa. And we always had, for this experiment, they had one glass tank and one other cage connected so that they had a large and more enriched environment. And we kept them in 2 conditions, either 2 females. These were sisters from the same litter, so they were the same age and they were nearly the same body mass, and they were highly closely related to each other by 50% and knew each other for all their life, and another male from another pair, or we kept them simply in pairs. So that's the control, and that's the experimental group. And what we found is that in these cases, in 37% of these communal groups, there was some aggression between females, and 33%, 1/3 of these groups we had to separate because one female was getting injured by the other female, and if you wouldn't have separated them, she might have injured the second female quite heavily or even killed it. While in pairs, only in 5% of the pairs, there was some aggression between the male and the female for whatever reason, and we had to separate the pair. No, we never had to separate the pair. In 5%, there was some aggression. We observed something, especially at the beginning when you put them together for the first 1 to 3 days. There can be some discrepancies, but then after this, it's all settled and there was never any injury and we did not have to separate them. And for the 2/3 that we kept together, there was infanticide observed in 39% of the groups. And this is seen here in this graph. So for way more than 1/3, so 39%, there was indication of infanticide. And this was probably by one female to the other. Infanticide was observed normally in the way. And we were weighting the females regularly, nearly every day. And then we might have seen a female was weighing 55 grams and the next day only 40 grams. So she gave birth, but there were no pups. So she gave birth and the pups disappeared because when they show infanticide, they normally eat the pups. That was normally the case that we found it. It also occurred to a low percentage in the pairs, and this is quite normal in rodents that infanticide can occur, that if they decide for whatever reason, maybe there was it was this under half natural conditions. Maybe there was a storm outside that disturbed them and then they showed infanticide. So this shows that there is high competition between females for reproduction. First, there's a high level of aggression that leads to separation of groups. And when groups are maintained, there's a high chance of female infanticide. And in fact, the communal pairs where the females were giving birth the same night, where there was synchronous birth giving, were the ones where it was the least likely to observe any infanticide, because when a female gives birth, she's maternally motivated and she won't show infanticide. And it's actually believed that synchronous birth giving evolved in many of these species, including striped mice, house mice, and zebra mongooses, to avoid the risk of the pups getting killed by another female. Benefits of group living. Why do animals live in groups? Can be increased vigilance. Vigilance, probably it's in French the same word, I guess. Vigilance means that they are aware of any potential predation risk. For example, goshawks, so these are some birds of prey, are less successful in hunting pigeons when they are in a larger flock. This is shown here, the flock size of the pigeon and the success rate for the goshawk. And if it's a single pigeon, it has a very high success rate. And if it's a large flock, only 1 out of 10 attacks will be successful on average. An ostrich spends less time in scanning for predators when it's in a group. So here you see the ostrich, it has his head high up and looks around for predators, but otherwise it's feeding. So they were measuring how often is the head up looking around for potential predators, which is of course not very productive behavior, contrast to the time the ostrich is feeding and they might not be aware of predators. And a single ostrich is much more vigilant than an ostrich living in a small flock of 3 to 4 individuals. Here, let's see. This here is a video of a nest of striped mice. We made this video in the field in South Africa. You see here white is some tissue paper, like this. We put tissue paper in front of the nest, and of course it's nice and soft. The mice take the tissue paper, carry it inside the nest. The nest is inside a shrub. And normally it's very difficult to see the nest because it's very well camouflaged. But if they take white tissue paper, then it's much more easier for us to find the nest because the white material is just shining. So it's nice and cozy for the mice and easier for us to detect. And I put a camera over the nest to video record with infrared during the night. And because these mice are diurnal, they are active during the day, they sleep at night. And what you see here is a time-lapse recording during the night. You see at 7 o'clock, it's not very dark yet because the sun didn't go down yet. Now at 8 o'clock, the sun is down. There's some infrared recording. You see the mice are forming one ball. They're sleeping together to keep each other very nice and warm. And very soon now you will see the ball is opened up here at 23 o'clock at night. It's like for you, it would be something like 2 or 3 o'clock at night. There must have been some disturbance. And we believe that living in a large nest like this, where not one individual is alone, but there's many individuals, there's always somebody awake. You see, even at 4 o'clock at night, somebody's always kind of awake. And that means there's always a sentinel who could warn the entire group if something is happening. And these nests are inside shrubs, at the base of shrubs in the open. And so there's a high risk that, for example, a snake, and snakes are some of the main predators of these mice. A snake is approaching the nest in the shrub. And then if one mouse can warn the others, then they could escape from this predator. And here now it's getting in the morning and the mice are getting up, going in front of the nest first to sunbask, and then they leave for solitary foraging. The delusion effect, imagine now with Zoom, it doesn't make a lot of sense, but imagine we would have been in a lecture hall and you're alone and the lion comes in. What are your chances to escape the lion compared when you're not alone in the lecture hall, but there are 50 other students and the lion gets one of us? Then obviously your chances when there are 49 other people is much higher for you to escape than when you're the only one he could find. And the confusion effect, this is one of my earliest studies I did. The confusion effect was always very important for people to explain anti-predatory effects of group living. The idea here is that for a predator, it's more difficult to catch one out of a group of individuals than to catch a single individual because he has to concentrate on the individual to get. And I tested this here with leopard geckos, and he got a small petri dish with some mealworms you might see here. And if I put one mealworm in there, they took only a few seconds and then they snapped the mealworm and they ate it. But if there were 20 mealworms, so quite some movement, all these mealworms running around, then it took the individuals much longer. So every line are the data of one individual tested under the two conditions. Every one of these geckos then needed much longer to catch one of the first mealworms to eat it. And also the number of fixations, that means the gecko looking around at the petri dish was very low when there was only one mealworm, but when there were 20, he was looking here, there, until he finally chose one. So this was one experiment to show that the gecko is kind of confused. Of course, in this case, the mealworms didn't have a chance, but this is also what you would like to have maybe in controlled experiments. The question here was, is there indication that there's a cost for the gecko to catch the food or not? And there is a cost. And under natural conditions where the mealworms might not be constrained in moving away, in the meantime, it might have disappeared within the soil. And I did nearly exactly the same experiment, but not with geckos, but with here these common marmosets. But it's basically exactly the same design because I was studying these marmosets at the time. They got a little dish with they for whatever reason they prefer the beetles that come out of mealworms over the mealworms. I gave them beetles, and here again, when they had 10 beetles, it took them much longer to catch one beetle than when there was only one, indicating that all these animals quarreling between each other confuses them and has a cost of time to catch this individual. There has been lots of discussion of group defense. Here you see these buffaloes living in herds where the adults, especially the males, are able to fend off lions. Or here these monstrous ox in the tundra building this ring of individuals with their horns outside to fend off wolves. So, and apart from all these benefits of predator avoidance of living in groups, there are benefits related to foraging, like finding good food sites, like clumped food, like fruits. This has especially been discussed in species like elephants and chimpanzees. let's assume the older, more experienced individuals will remember where there is a seasonal food source and will be able to guide the entire group to this food source. Or also, especially in elephants, the water sources. It's also been discussed that colonies in birds could function as information centers, and that individuals that were not very successful in foraging, when they come back to the colony and as they see another individual was very successful, that they will follow this individual back to the foraging grounds to find out, for example, where there's a big swarm of insects they could feed on, or some other areas with a lot of food. Cooperative hunting has been discussed a lot in the 70s of the last century, millennia, especially this cooperative hunting like you see in wolves or in lions, because it's very impressive for us to see how a pride of lions is hunting, but it was then soon found out that this is not a very big benefit and that they do not really have good strategies. I have observed myself lions many times in different areas in Africa when they are approaching together a zebra to hunt, and normally one thinks, oh my God, they are so stupid because they don't really coordinate their actions the way we would like to see it. And the benefits of cooperative hunting are probably not really very important. And there's been many studies. There have been millions of research funds giving from the military to scientists studying cooperative hunting in large carnivores, because the military thought, or maybe they have evolved strategies that are very clever, and understanding these strategies might help the military in the US to have plans against invading Russians. But after 5 years, the military stopped giving money to the scientists, and animals are not very well in coordinating their hunting efforts. Sometimes it works. Here we have hunting success of jack. So that's a fish, a large predatory fish. And prey hunt captured per individual increases from 1 compared to 3 or 5 individuals. There's no difference here. So there can be some benefits. And the benefits of cooperative hunting are typically always due to simply the presence of several predators, but it's very rarely that there's coordination, that different predators take over different roles. It's simply like these wolves are running behind the deer, and if the deer runs away and the wolf gets very tired, there's just a chance that there's another wolf close by who is not that tired yet. And so the chance of the wolves increase and of the deer is decreasing. This is again the same, the check. So the hunting, the chance of hunting success increases when you're in a larger hunting pack, hunting school in this fish. And when you're the first fish in this hunting group, then your chance is even higher than of the other fish that are swimming behind you. Cooperative territory defense can be a benefit, that you might be able to defend a territory when you have many coalition partners, like in a baboon group or if meerkat group, compared if you would be alone. And other benefits that can be quite important have been overlooked for a long time because they might not be so important for large mammals or large species, are thermal advantages of being close together. It means that individuals warm each other. These are thermoregulatory benefits. And you have seen this video before. And you can imagine for a mouse that lives in a nice ball together, they keep each other very nicely warm. And this can have significant benefits than when the mouse sleeps alone. We studied them in South Africa in a semi-desert where even in summer, temperatures can go down to 5 degrees because the sky is open and the heat just disappears. And we measured their oxygen consumption. This is their respiratory rate. So this is a measure of how much energy they spend. The more oxygen they burn, the more energy they spend. So this is, where's the mouse? Milliliter oxygen per gram and hour. And the higher it is, the worse. And it depends, of course, on the ambient temperature, because at 30 degrees, it's a thermoneutral point. 30 degrees, they don't have to spend any extra energy in maintaining their body temperature. The basal metabolism is doing the job. But if it's colder, 10 degrees, very common even in summer, even 5 degrees, common in summer, in winter it can get even lower. They have to spend much more energy. You see it's increasing by the factor of nearly 6. And this is especially the case in the mouse that we kept in these chambers. We kept them in a chamber and we slowly increased temperature, and this decreased the oxygen consumption. And the solitary mouse always spent much more energy than any mice living in, not living, but sleeping in a group where they could warm each other, showing that they have big benefits of this behavior. So for the evolution of group living, the costs of it have to be lower than its benefits, but the costs and benefits to vary with varying environments. And this is then the research field of social ecology. If it would be the same for every species, it doesn't matter what the environment is, and all of them would be living under the same form of social organization. The high biodiversity of social organizations we observe in the animal kingdom, often even between closely related taxa, indicates there must be factors determining whether the benefits are higher or the costs are higher. For example, here we have these cliff swallows, Hirundo where we have these costs of ectoparasites. I told you before about what the benefits of that it works as a colony information center, where an unsuccessful bird can see a successful bird. It comes back to the colony and follow it back to where there might be a swarm of insects where there is sufficient food to be found. And here we have these costs again of these bugs per nestling, and we see again there's an increasing cost with increasing colony size. And this also has an influence kind on the body mass. I mean, you have a benefit, you live in a larger colony, but this is then compensated by the costs of having more ectoparasites and the nestlings. So the nestlings are the chicks when they are ready to leave the nest. No, that's not correct. That would be fledglings. So these are chicks in the nest, and they do not benefit. And even at a much larger colony sizes, the nestling mass is decreasing. Now, what they did here is the idea was that it's not increasing further on because there would be benefits, but the costs of the ectoparasites are increasing. So they were treating these nests with an insecticide to kill the bugs, to get away of all these parasites. And then they found that the picture that is very indifferent here showed a clear trend that with increasing colony size, there was a benefit in the form that the nesting mass was increasing. And this shows the trade-off between the costs of ectoparasites and the benefits of living in a group. And here we find that this trade-off is kind of kept constant, and this can explain why we can observe these colony sizes of different sizes, because it doesn't make a difference if the colony has 50 individuals or 100. Would it be 122? There's nearly no fitness benefits because the benefits obtained here from a larger group are compensated by the increased costs of having more parasites in the nest. Another interesting study was done with these freshwater fish, the guppies, in I think it was in Trinidad in Middle America. And these fish, they form larger groups for predator avoidance. They were taking fish from a river with high predation pressure. And so living in a group has benefits. They see the predator early and can escape it, but there's increased competition for food. Then they took some guppies and put them in another river system where at the top predatory pressure was very low compared to here, and at the bottom it was the same. And this here is an area I don't even remember, but this is many kilometers. It's maybe something like 50 or even 100 kilometers from here to there. So they released the guppies there, and then over the years, and there were before this, these fish did not occur in this river system. It's a study you couldn't do these days anymore. It's also a study from the 70s or 80s. And then the fish were migrating down there, not migrating, they were reproducing here and the offspring were dispersing. Okay, that's 30 years. And what they then found is that the fish released here, they were forming much smaller schools because the benefits of predator avoidance were here not that high anymore because predation pressure was much lower. But the fish, their descendants that dispersed down here were again forming schools as large as on the original population because here the population pressure was similar to there. Good. So we have again the socio-ecological model of social organization with the distribution of resources and predation risk determining the outcome of the cost-benefit calculations for the females, influencing female distribution. The risk of infanticide might have another effect here. And then the female distribution is determined. Do they live in groups or do they not live in groups? And this then influences the distribution of males. And when males are living in a group, then they might in addition increase their reproductive success, their fitness by showing paternal care. So in other words, the distribution of resources in form of food and shelter doesn't have a strong influence normally on the distribution of males, but for them, it's the females that are the resource influencing the distribution. So now I would make a break of 10 minutes until 3 o'clock so everybody can get a break, a coffee or whatsoever. It also means if you do have a question, I'm going to make myself a coffee. If you have a question, you can write it in the chat function, both here in Zoom, where we'll especially look at it. Is there anybody on Facebook? I will check if Facebook is more difficult for me to, not Facebook, YouTube. There's only one person. So if you on YouTube, you have questions, you can also write it here. So I'm going to continue now. And want to show some examples how the female distribution can be related to the male distribution. Again, on my own study species, the striped mouse, Rhabdomys pomelio from South Africa. And we did a study in the grasslands where resource availability is actually for food is kind of lower. Resource availability for food being lower means the females, which are shown here in dashed lines, they cannot share a territory because there's little food. There's lots of green grass, but mice are not cows. They cannot eat grass. They eat seeds, they eat herbs. And within this grass, this field, within this sea of grass, there's not so much food. And we see the females are all alone. We radio tracked every single female here and all the males. The males have large home ranges. These are the solid black lines overlapping the home ranges of several females. In contrast, here in the succulent karoo, and here we radio tracked only about half of the mice, we see that they are forming groups. You see the mice with the same color, like here, these violet mice, the red mice, the black mice, the yellow mice, the blue mice. They overlap to a large extent, and here every group of females has one male that comes from another group and is the breeding female here. And also important to notice that the scale here is quite different. So the area shown here is much larger than the area there because of low resource availability. They need larger home ranges. So females live in communal groups in the succulent karoo, while females are solitary in grasslands. And the prediction that we made, or the assumption, is that the males follow this distribution. When the females become solitary, they become solitary with large home ranges overlapping several females. And if they form groups, males try to monopolize groups. And these predictions became testable in 2003 after severe drought. Survival probability decreased in this year. Survival probability of adults at the beginning of the dry season to survive till the next breeding season decreased from 27% to only 1%. And population density declined dramatically from 32 mice per hectare to only 1.5 mice per hectare. And afterwards, after the breeding season, it increased a little bit. And with this decrease in population density, females change from group living to a solitary lifestyle. What we see here is the data from the succulent karoo, where they were group living before, for 2003, which is after the drought. And here we see the female home ranges. They are all solitary, and black, the male home ranges that are very large and overlap the home ranges of several females now, which, if you only look at it from a broad perspective, looks much more similar to what we found in the grasslands the year before compared to what we found in the succulent the year before. And again, this area now is much bigger. This violet quadrant is the entire field site here. You see here in this quadrant, we have 2 females and 2 males partly, while in the year before in the same area, we had nearly 60 mice living. So that showed us that when the females switched here, the succulent karoo from group to solitary living, then the males also became solitary living, increased their home ranges. The alternative, of course, could have been that they could have formed pairs. One male could have associated with only one female, but with this, the individual male would have decreased its mean chance of reproductive success to reproduce with more than one female. The home range sizes also developed accordingly. What we see is in the pre-breeding season, home range sizes were quite small. The females remained the same home range size about throughout the season, from the pre-breeding through the breeding to the post-breeding season, while males, when breeding started, they increased them dramatically by the factor nearly 5. Why? Not because they needed more food. Actually, in the breeding season, is after the rain, there was more food, but because with increasing the home range, they obtained potential access to more females than before. So this showed you already that within one species, there can be intraspecific variation in social organization, the solitary mice in the grasslands compared to the group living ones in the succulent karoo. But even within the population of the succulent karoo from year to year, there can be variation from group living to solitary living. And this intraspecific variation in social organization, for this we use the short name IFSO. So IFSO is important to understand the social system of a species, because it would be wrong to say striped mice are group living, because often they can live solitarily. And if we ignore this potential of solitary living, we would miss an important part of their social system. It can be a tool to study the proximate and ultimate reasons of sociality. I cannot bring this here in detail, but I think I did in previous lectures. For example, we can compare between solitary living females and group living females, whether there are differences in hormone levels. I think I showed in the lecture on alternate reproductive tactics that females that are solitary living have lower corticosterone levels. It can be part of the answer to the question, will populations be able to cope with global change? When the environment is changing in species, populations might have to change their social organization to be able to cope. And so the question whether or not they can show this change in social organization can give us an idea about their resilience to change. And importantly, presently, the phylogenetic approach to understand the evolution of the variety of social systems is flourishing, and this needs the correct classification on the species level. So phylogenetic approach, I will talk about this in more detail also tomorrow, is we compare different species living under different environmental conditions. We know what is the social system, and we want to see in how far does the environment versus the common ancestry explain the variation in social systems that we do observe. And these studies, of course, can only be done properly if we classify the social organization or social system, tomorrow we talk about care system, correctly. If we classify it wrongly, then we might not be able to do a proper study. And importantly for me, IFSO is itself an interesting phenomenon to see that these species, I couldn't, when I started these studies on striped mice, I couldn't believe it, that they can switch from solitary living to group living and back. This is an interesting phenomenon by itself that needs scientific explanation. So in theory, there are 4 different ways how IFSO can come into existence. It's shown here in this graph. And this depends on whether it occurs between or within individuals and whether it is explained by individual plasticity or not. In these 2 cases on top, we observe IFSO because different individuals behave differently, which can either be due to genetic variation. These individuals here are genetically different from those individuals. This would not be any plasticity in their behavior, or it could be due to developmental plasticity, which is non-reversible plasticity early in environment, where in environment 1, they develop a different behavioral phenotype than in environment 2, leading to different forms of social organization. Or it can be within the same individuals. The same individuals might at one time be pair living, another time they might live solitarily. And this could be not due to them changing their behavior, but something happening to them, which is what we call entirely extrinsic factors. For example, in pair living species, if one individual dies due to predation, due to old age, out of a pair, if one is gone, there's only one individual left, and we might observe variation that is non-adaptive and imposed on the individuals. Or here we have the case of the striped mice. You see a variation between the same individuals that can switch from group living to solitary living and back. And this would be an adaptive response that we call social flexibility, which is reversible phenotypic plasticity of the same individuals. So the question we have to ask if we want to find out which one of these 4 mechanisms is important is, occurs it in different or the same individuals? Is it due to some kind of plasticity, reversible versus non-reversible, or is it due to something else happening? So the first case genetic variation would be that there's no individual plasticity and the variation is observed between individuals. So different, and we might have, for example, different populations living in different environments, different selection pressures, leading to local adaptation of the populations, which then show different forms of social organization. And this we would observe. Population 1 lives in groups, population 2 lives solitary or in pairs. That would be intraspecific variation in social organization. And this, for example, has been described to some extent in prairie voles, a small mammal from North America, often used as a model to study monogamy and pair living. But some of these populations, like here in Illinois, are typically found to be pair living, while in other, oh, these colors are wrong. This here is, why are these colors wrong? So this here is Illinois, where they are typically pair living. And here in Kansas, they are most often found to be solitary living. And there have been different studies on the genetics and especially the genetic regulation of physiological mechanisms of living in pairs. And some factors have been discussed, like the ER alpha. This is estrogen receptor alpha. It's a receptor for estrogen in the brain that modulates social behavior, and there were some differences found. Or the arginine vasopressin receptor 1A and its genes, and there has also been some indication found. And I quickly want to show one of the newest and most sophisticated studies that was done by the groups of Stephen Phelps. But all in all, I have to say there's very little evidence that IFSO is explained by this mechanism, but as it's interesting, I still want to talk about it. It's, like I said, the research group by Stephen Phelps at Austin University in the southern United States, and it's a paper published in Science 5 years ago about that should all be orange, the vasopressin 1A receptor, which is written like this, and the gene encoding this receptor is written with small capitals and in italics. So when you read any papers on this, it's sometimes confusing because it's nearly the same, but it's this is even wrong. It should be V1RA. But one has to differentiate whether they talk about the receptor or the gene encoding for it. See here, this is the gene. I have to look if I did something wrong here. Okay, so what they found here is this is some, they were studying them in big enclosures where they put several males and females in, and they differentiate here between two types of males. These are the the males with high sexual fidelity. These are pair living males. And then there are males that have less sexual fidelity and that are wandering into the neighboring territories trying to mate with additional females. And what we see here is that these home ranges and how they are used are differing between these males. So they are very restricted to their own home range and their own female, while this male that is an extra pair fertility male is also going into other areas. So they have also larger home ranges. These males that are looking for extra pair paternities, they then also are more successful in getting extra pair paternities, but at the same time, other males are more likely to intrude their territories because they have less time to defend them, and other males are also visiting them. And importantly, this is coming here, this is then related to differences in the expression of the AVPR1A receptor in specific brain areas, especially here. We see here, this is the RSC, the retrosplenial cortex. There's a lot of expression in the monogamous males, but in the ones with high extra pair paternity, this is different. And I cannot discuss this in too much detail here, but just to show you the different forms of evidence they found. They then did also studies where they found that these receptor expression, the genes for this receptor expression differ between these two types of males, and showing that as there are genetic differences, these differences could also be under natural selection, indicating that these two types of males are genetically really different, and they also found evidence that on average they might have the same reproductive success, explaining why both male types are maintained within the population, because while they are genetically different and show different strategies in their social behavior, they do not differ in evolutionary fitness. If they would differ in fitness, you would expect that one male type is taking over. And this is shown here. Here you have the relative fitness of the males that have the high or low expression of this vasopressin 1R receptor. So these would be the socially monogamous males, and these would be the males that look for extra pair fertilities, and the reproductive success on average is about the same. So this is the best example I have where genetic differences could influence the social system and maybe the social organization. But all in all, there is not much evidence that genetic differentiation is an important mechanism to explain observed ifso in the field. To sum up this study, males differ in ranging behavior. This correlates with success in gaining extra pair paternity. The males that show these excursions, they have higher extra pair paternities. And this is also related to differences in the brain. The V1AR receptor, and this is represented in differences in the genes regulating this receptor. But why is this like that? But I only added these slides. I should have checked it again. But this is not related to fitness differences. If there would be fitness differences, you would expect one male type to take over. So the second kind where ifso could be explained is by entirely extrinsic factors, where variation is within individuals, but it's not due to plasticity. And this is a mechanism that is quite common in nature. And what we observe here, there are environmental disruptors that leads to a non-evolved social organization, and then there's an individual response. So the change in social organization is not due to an individual response, but the enforced change, for example, due to the death of an individual, then leads to an individual response. And this individual response will allow the return to the evolved social organization, the social organization with the highest mean evolutionary fitness. But there might be different groups, and when somebody like a PhD student goes to the field and observes several groups, there might be different stages of this environmental disruption, and then the student might observe two forms of social organization, and from this conclude that there is intraspecific variation in social organization in the species, as at one moment in time more than one form of social organization is observed. To explain this in more detail, I take a theoretical example of common marmosets that are said to be pair living, that have breeders, one male, one female breeder typically in the group, and adult helpers of both sexes, often their offspring. And so what we might observe normally is one breeding male, one breeding female, and adult female and adult male helpers. But now the Tyra comes. It's a mustelid that likes to eat little monkeys. These monkeys are only that size. They are, I think, around 500 grams, so they're not very big. And now let's say the Tyra eats the female, and obviously now the social organization looks different. And there is a vacant position for a breeding female in the group. And these adult females in the group that so far did not reproduce might compete with each other about who is gaining this breeding position. Then we might see at one time that not only one, but actually three females in the group are breeding. They might still try to expel each other. They might kill each other pups. But for this time of observation, you have a group of one breeding male and three breeding females. And then the males might also start to compete with each other. This could lead, for example, that another female from a neighboring group that's stronger and more competitive immigrates and evicts the other females. Now the males are fighting with each other until one of the male wins, and then we come back to the social organization of pair living with one male and one female. But in between, we find all different forms of social organization. but this is induced by the external disruption of the predation event. And this change that has been induced here then leads to adaptive responses of males and females competing who will take over as breeders. So in other words, this kind of if-so that occurs quite often in nature is not something that has evolved, but something that is enforced. And so we don't need to explain it from an approximate or ultimate point of view. What is then interesting to study are the evolved mechanisms of female and male competition about the breeding position, but not the if-so per se. The third possibility is developmental plasticity. And developmental plasticity is quite common in nature. It's an adaptation to predictable changing environments. We have an individual response to the environment, and one of two or more potential phenotypes is developed by the same genotype. So individuals living up in environment 1 will develop a different adult phenotype than individuals growing up in environment 2. And it's always adaptive, meaning that they, with this developing the specific phenotype, they have the highest reproductive success. And developmental plasticity is very common in nature. It's observed, for example, in tadpoles that live in an environment with predators, like here, this is this larvae of a salamander. They develop differently than tadpoles living in an environment without predators. In the predator environment, they will develop a much bigger head, so they are have a lower risk to get swallowed because they just are too big. They might also develop much faster to escape from this predator high environment, but tadpoles of the same species, even of the same parent, raised in a pond without predators, they will develop a small head, they will grow longer, will grow bigger, and leave as bigger froglets the pond. And something similar, I think there's here you see the difference between a tadpole. They're genetically the same, growing up in a high predator environment versus low predator environment. And similar things have been observed in Daphnia. When a high predator environment, they develop a phenotype that is very spiky and spiny. So it's not so easy for a predator like a fish, a three-spined stickleback, to eat it. While if there's no predator, they don't invest energy into developing these kind of anti-predatory defenses, and they look very different, but they're not genetically different. So developmental plasticity is common, it's adaptive, but I do not know of any case that explains if so. And I think the main reason is if there are predators in the environment, a quick response to develop phenotype 1 or 2 has a big benefit with regards to fitness. But to determine early in development under which strategy you will breed later, it might be much more difficult to achieve because it's difficult to predict how the environment will be in many months, in some species many years, when the breeding stage is achieved. While if you're a tadpole, you can immediately predict whether predation pressure is high where you're living or not, and then determine the best phenotype. And then last but not least is the example of social flexibility. And I showed you the example of striped mice. This is then the most common adaptive form of if-so we observe in nature. So individual response environment A, the genotype produces the behavioral phenotype A, the environment changes to B, and then the genotype produces the alternative phenotype B. And this phenomenon is that the social organization of an entire population can change as a function of individuals of both sexes changing their reproductive tactics in response to changing environmental conditions is called social flexibility. In other words, here we have again, like I told you 2 weeks ago, alternative reproductive tactics. But importance here is we have alternative reproductive tactics simultaneously in both sexes, both males and females, not only in the males. And if both sexes have these alternative tactics, then it can change the entire social organization of a population. And this has been observed in many species, from invertebrates to vertebrates, like these burying beetles that can form pairs that can form one male multi-female groups or multi-male multi-female groups, depending on the carrion size on which they feed. So these burying beetles, you might know them when there's a dead animal, like a dead mouse, they find it and then they dig a burrow where they put the dead body in and they raise the offspring there. And if the mouse is very big, it can be enough food for the larvae of 2 females. If it's small, maybe of only 1 female. This has also been observed in the prairie vole, which I talked about earlier, that might not only be living in pairs, but also in communal groups, sometimes also in groups where more than 1 female is breeding, in house mice, in Danox, Prunella modularis, in the gray kingfisher, and of course in the African striped mouse. And so this allowed us in striped mice to study why animals live in groups, but also why do they live solitarily. Typically, one of the main hypotheses of why animals are living in groups are ecological constraints that predicts that when population density is high and all territories are occupied, individuals are forced to stay in their natal group because they cannot go anywhere. There are no free territories. And this has been shown experimentally in different species like this fish, Lamprodocus brichardi, the Seychelles warblers, or also the prairie voles. And the second main hypothesis here has been reproductive competition, saying that competition for reproduction between groups induces costs of staying in a group, and this would favor solitary living. So if there's a high reproductive competition, to avoid the competition, the best is to leave the group and to become solitary. And reproductive competition has also been found in many, many species like meerkats or house mice. There then often leads to female infanticide, but this is then called reproductive skew. And normally you can observe this competition between groups, but it does normally not lead to solitary living. Now in the striped mice, which you already know, they can, and I showed you already, they can either live in groups. Every circle here would be 1 group with 1 and 2 or 3 breeding females, or they can live entirely solitarily. And it's nice to study them because it's a diurnal species. That means they're active during the day, in contrast to most other mouse-like rodents, which are normally nocturnal. And large sample sizes are there possible. We can observe many groups at once. At the field station in South Africa, we had been studying typically 8 to 12 groups with up to 200 individuals at the same time within a relatively small area. If you would want to study 12 groups of chimpanzees or lions, this is nearly practically impossible. We can do experiments in captivity to study things we can't see in nature, or to have experimental approaches. Important. We also like to do ecophysiology to measure hormones. We can easily get blood samples of hundreds of individuals. For example, here, this is a method where we get blood from the tongue. Most people, when they took blood earlier from rodents, they took it from the eye, which can be quite critical. And there's a new method to get it easily from the tongue. And importantly, for evolutionary questions, it's a short-living species. Their 1 year represents 1 generation. They're breeding in spring, which in the southern hemisphere starts around August. So they're breeding from August till November, December. Then the mice born here have a chance between 1% and 30% to survive the hot, dry summer and cold, wet winter, and then reproduce here. The summer can be very hot, up to 40 degrees. It's very dry. There's very little rainfall and very little food. Then rainfall starts, depending on the year, around May, June. And then it's raining. It can also get quite cold, quite energetically demanding. But after the rain, there will be more food again. And what we found in one of our first studies with data from 8 years of observations of 1 population is a relationship between population density and sociality in the breeding season. When population density is high, nearly all mice lived in groups. When it was very low, that's the year 2003, I had shown you before, all mice are becoming solitary living, and in between we found both tactics expressed by different individuals. And interesting, and this we used as evidence that ecological constraints in the form of population density in free territories constraints group versus solitary living. The higher the population density, the smaller the home ranges, which we also found and the more mice live in groups, supporting that ecological constraints favor group living. Now here we added data from the non-breeding season in winter and summer. So this year is always data from September. Pre-breeding is, depending on the year, July or August, and post-breeding is normally January. And just by the way, the mice here are exactly the same mice like the mice down there in the year 2003. This is something like July, and this here is September. And what you see is that all these mice nearly all these mice were group living, and all of them switched to solitary living, also showing that they had the option to live in groups if they would have wanted to. It's not because there were no mice left to form groups, but all of these were living in groups 2 months before. So what is different? So there seems to be a difference between the breeding and the non-breeding season. Obviously, this is whether or not they breed. And so our conclusion was that the additional costs of reproductive competition during the breeding season makes them live solitary living if population density is low and free territories are available. Without these costs of reproductive competition observed here, they nearly always live in groups. And we also tested this experimentally. This was a PhD thesis by my student Ivana Schöpf. On a neighboring field site, she had different groups. So every polygon here is the home range of one group. There were some R groups, removal groups. The mice here were trapped and taken away. And the neighboring groups, E1, so this is E1 twice, so this is one replicate, were predicted to move into this area, these sub-individuals to start solitary living because we provided now free territories, while the control groups that were not directly neighboring there experienced only a low reduction of local population density. So these mice were removed, and we expected some mice to move in there to become solitary. And this is exactly what she found, that the percentage of solitary mice was much higher for these mice that had access to free territories than the other mice. And this was only the case in the breeding season. When we repeated this experiment in the non-breeding season, even when there were free territories available, they remained group living to benefit from benefits of group living, because there was no benefit of starting your own territory. And these are the data from this experiment. And you see the curve here, though it doesn't go as far down like here. It looks very similar what we found from the experimental approach compared to the descriptive study. Now, after this, the last years we wanted to see, what we see here is that within one population, if population density varies, then sociality varies. And the next question was, is this social flexibility also an adaptation not only to variation from year to year, but maybe also variation in space? And for this, we studied within the Guchab Nature Reserve 8 subpopulations. Every blue circle here is one subpopulation. You see this here is 4 kilometers, and they were separated by sandy flats or by mountain passes where there are no striped mice living. And we studied the population density and the sociality. And these are these black data. And we found exactly the same pattern, or nearly exactly the same pattern, compared to what we found before for 8 years of the same study population. So showing that not only variation from year to year, but also from subpopulation to subpopulation induces this form of variation in social organization. That means how many mice are living in groups and how many mice are living solitarily. So what are costs of reproductive competition? I showed you this graph before. For females, it's the risk of aggressive female-female interactions and female infanticide. And we think that to avoid these costs of female infanticide, some females decide to move out and start independent breeding if free territories are available. We also found costs for the males to live in groups with studies on the captive colony then at the University of Zurich. And here we kept males in families or solitarily. So we had family groups living here. And when the offspring, they are weaned at 2 weeks of age, so they don't get milk anymore. And we separated them at 3 weeks of age. We had one male who remained with the family, and his brother, who was the same age, everything, we put him solitarily, because in the field this is the earliest age they might leave the group to start solitary living. And what we found is that the males that are kept solitary, they went earlier through puberty. They have much higher testosterone levels. We see here the testosterone levels at the end of the study. The family guy has very low levels compared to his brother. And they have lower corticosterone levels. It's a stress hormone, and much larger testes. This here is the testis mass. The testis of the male who was put single was more than double as big than compared to his brother who stayed with the family. So all of this indicates that there's sexual suppression happening in the family to the males that remained there. And we repeated this study with both parents present, only the mother present, or only the male present. And this result was only found when the father was in the group. If you remove the father, then the males in the group developed in exactly the same way like the solitary kept brothers. So population density and reproductive competition are important for the expression of social organization in striped mice. And there's indication that in other species with social flexibility, that's also the case, like in the burying beetle. With high population density, you're more likely to find groups. And also when there is less competition, when the carrier and they find the food is big, so there's less competition because there's enough, then you are more likely to find larger groups. The same in the gray kingfisher. Population density has an influence whether there are hairpos for a pair or not. And reproductive competition, here's the form of nesting sites. They build nest burrows in steep walls of soil at some lakes. And if there are lots of nest burrows, then they're more likely to form simple pairs. But if there are few nest burrows, it means there is competition for this reproductive site, they're more likely to form groups. Population density is also important for the prairie vole and reproductive competition, and also that's also the case for house mice. So it seems that in general, population density and the degree of reproductive competition has an influence on the expression of variation in social organization in a wide range of species. Now I come to this point I mentioned at the beginning, that if so, it's not something we just say, okay, it's some variation we don't have to care for, because we think it's important when we want to do comparative studies where we compare many different species, that it might not be good to put them into one category, but to say, okay, they're all in pair living, they're all living in groups, but that this variation should maybe be taken into account. And so we started a comparative database on social organization in all mammals and recorded everything that's known from field studies. So studies where these animals have been studied in the natural environment compared to captivity. Because in captivity, if you put one male and one female together, you have a pair. And that works in most mammals. And it doesn't mean that this species is pair living under natural conditions. And the first taxa we did this for were the Eulyptotyphla, which were formerly called insectivores, but then there was some change in phylogeny, I don't know what, and now they're called Eulyptotyphla, very difficult name, and these are the shrews and their close relatives like voles, for example. And we compared our results with a large database on mammalian social organization used by Lucas and Glattenbrock in very high-ranking papers like in Science and Proceedings of the National Academy of Sciences. And we found quite differences, because 7 species to be solitary living, 4 to be group living, and 5 to show if so. While they found 397 species, and these are only the shrews, not the other relatives, to be solitary living, 2 to be group living, and none to have if so. But interestingly, if we compared our results with Mammals of the World, which is a very big book written by experts in the field, they found very similar results to what we found. And So the problem here is they did not take if so into account, and the other problem, they have a lot of data of species that have never been studied in the field, because only these, what is it, 16, only these 16 species have been studied by zoologists in the field, and the remaining 381 have never been studied. But Lucas and Clattenbrock assume that shrews are generally solitary, and thus they entered all these data into the database, and we think it's wrong first to enter data for species that have never been studied, and not to consider variation in social organization. And why is this wrong? You see, if we take the approach where we only use the information that's really reliable, we find that 44% of species, so not even every second one, is solitary, while they find that 99% of the species are solitary. And why do they find it? Because they assume for 381 species that it's a form of social organization. So they find their own assumption. And one of the first studies published in the year 2007 was by F. Dalerom. That took if so into account was on the evolution of social organization in carnivores, where previously, like for all mammals, for whatever reason, it was assumed that the ancestor was solitary living. But if he took the flexible form of social organization into account, then this result was not found anymore. So here these circles show what is the probability that the ancestor was solitary living. It's the highest, but it's less than 50%, or flexible or something else. And for some taxa, like the Canidae, so these are the dog-like carnivores, the most likely ancestral form was in fact found to be a flexible form of social organization. And we did similar studies like this, where we really looked only at the primary literature, and we took if so into account on the evolution of social organization in primates. It was by a master student here in Strasbourg, Camille Pilisi. And she found, when we take only this information into account, in fact, the most likely ancestral form of all primates was not solitary living, as long believed, but variable. And solitary living, which people assume to be the case simply because one knows it without data, that the ancestor was solitary, and we found nearly no support for this. And I don't show it here in this lecture, but within this variable form of social organization, solitary living was the least important component. So, and we are building up this database at the moment now for all mammalian taxa. I hope we will finish by the end of this year. We are making good progress. We have here different taxa, like the opossums, carnivores, marsupials, elephant shrews, armadillos, shrews, and the bats are nearly finished, whales and dolphins, and so on. And we only, like I said, we only take information into account when the species has been studied in the field. If somebody puts a male and a female together in the zoo, we do not believe this is proof that they are always pair living. And the interesting thing is that for most of these taxa, very few species, in fact, have been studied in the field. Like from the opossums, only 8 out of 98. And in the primates, it looks a bit better. And in the even and odd-toed ungulates, it also looks quite good. But then in bats, there's very few species studied. In shrews, the Olyptotyphla and their relatives, very few, and so on. So we really miss a lot of information about what's really happening in nature. And interestingly, if so, is very, very common. Originally, we started this project to see when does if so evolve. So we expected some species show it, a few species, and then we want to know what is special about these species. But now with the exception of the opossums, where very few species have been studied, and what is this? The armadillos, which are mostly solitary. We find that if so is actually very, very common. Here, the odd-toed ungulates also have a low. And we now might have to change our research aim, not to explain why does if so occur, but why does stable social organization occur, because a stable form of social organization seems to be much more rare than a variable one. And also interestingly, solitary living is by far not the most common form of social organization in mammals that really have been studied. It's quite common in opossums, but few species have been studied. And I predict that it will change here when more species get studied. But in all the other taxa, it is quite uncommon. So I do not know why people, and including myself, normally believe that most mammals are solitary living. And this might lead to a change of how we understand our own evolutionary history, because I grew up with this picture in my head that our mammalian ancestor that lived in the dawn of dinosaurs was a little nocturnal, solitary guy running around trying not to get eaten and then being very asocial. And only after dinosaurs got extinct, they evolved into something more sophisticated. But this might not be the case. And in fact, if we look at what other fields of research, of science have found, it raises the questions why we had, or at least many people had this assumption that mammals are by default solitary. Here we have data on cynodonts. Cynodonts are reptiles that lived 260 to 230 million years ago. They're reptiles, they're not mammals, but they are the direct ancestors of all mammals. And data from paleontological evacuations indicated that many of these species, or most of these species, might have been in fact group living. For example, they are normally not found solitary skeletons, but skeletons of groups or pairs. Like here we have this block of fossil, and you might know if you put up a block, you have a positive and a negative. And here we see different individuals indicated in this drawing of this fossil in different colors as a blue, a yellow, and a green individual. And fossils like this of the cynodonts are not the exception, they're the rule. You rarely find a fossil of only one individual. For example, here, I mean, this is how many million? This is also more than 200 million years old. This is a Trinasodon. They are 240 million years old. That doesn't look very solitary to me, this early ancestor of the mammals. They also found fossilized burrow systems of some of these cynodonts, and in these burrow systems, fossils of many adult and many juveniles living in there together, again indicating that they might have been already quite sociable, our ancestors. Then there's a big gap in the fossil record, and the next one that I found is of this marsupial from, I think it's Bolivia in South America. One of the first records of several mammals have been found, fossils, and here also they found for this marsupial, early marsupial from, I think it's 60 million years ago. Yes. They've also found together the remnants of many adults of both sexes, juveniles and pups, again indicating a gregarious lifestyle and not a solitary lifestyle. So there's evidence against a solitary ancestor of mammals. The first one, which one should not forget, is that by definition, mammals are highly sociable because all mammals show some degrees of maternal care. Because without care of the mother, the mammalian offspring cannot survive. Then fossils indicate a gregarious lifestyle. Gregarious means kind of group living, though we don't know exactly what was the kind of groups they lived in and how stable were they. In cynodonts, the reptilian ancestors of mammals more than around 250 million years ago, and of early mammals 60 million years ago. And our comparative studies we're doing here in Strasbourg indicate that most species can be group or pair living, and that solitary living of the species that have been studied is actually quite rare. And there are very few well-studied mammalian species that are truly solitary. Exceptions might be some big bears, the tiger, and very few other species. And this raises the question, was the ancestor of all mammals maybe social? Is solitary living a specialized form of mammalian social organization and not the ancestral form? And the next years, we want to focus also in our field studies in understanding why do mammals live solitarily and what are the costs of solitary living, the benefits, and what problems do solitary living animals, especially mammals, have to face. So this could lead to some transformative science, changing the way we see our own ancestry. Maybe it was not this little solitary guy running away from the dinosaurs, but that our ancestors were already quite sociable and had some form of group living that helped them to survive within this age of these very, very large reptiles. Good. With this, I'm finished of the lecture of today. There are 10 minutes left. It was quite a lot of information, so I don't want to start talking about social structure, but give you the opportunity to ask any questions that you might have, or if there's something I should repeat and talk about again. So you can either ask in person by just switching on your microphone, or you can type just your question into the chat function. And I think on YouTube there's nobody. So somebody asked me to do, but maybe some people will look at it later after they come back from their test. I don't know. Okay. Why does it show this now? Okay. So anybody has some questions? So you can still think about, use the time that you have. I want to summarize again while you are thinking. Social organization is about the composition of social units. It can be quite variable. And there are 4 potential mechanisms, of which only 2 are really important. The one is environmental disruptors, which is a non-adaptive imposed change. So we can't explain it via evolution or physiology. We can ask what happens after the change has been imposed, but not to explain the change itself. And the second common one is social flexibility, where individuals can switch back and forth between alternative tactics. And the reason why we think flexibility is more important than having a fixed alternative form is that once the individuals breed, this is long after they went through their ontogeny, and early in ontogeny, they might not be able to predict under which conditions they breed, which also means they cannot predict what would be the best tactic to show them, which is why being flexible is more adaptive. Then for tomorrow, I will use exactly the same Zoom link for tomorrow morning like now. There's no reason to make 2 Zoom links, so you can then switch on in this link. And if by then you have any remaining questions, then you can also ask. One question I have is, do you know why there are so few people here? I mean, it's only 18, and I thought it's nearly 60 or so in your course. Okay. Are all of you now in quarantine, or did all of you already get tested, or everyone is in quarantine? But you can go out and get tested, and when it's negative, then you're allowed to. Do you have, just out of interest, because even though I work in France, I live over the border in Kiel, I'm more by this. Okay. Do you have the 5 days rule that because after one is exposed, one might have it, but one might only be able to show it 5 days later? So do you have to have 1 test or 2 tests in France? Because in Germany, you would have to make 1 test now and then in 5 days again to be sure. And only then it's the same? Okay. So you're Tuesday, Wednesday, Thursday, Friday, Saturday. So Sunday you might be free again. No. Tuesday, Wednesday, Thursday, Friday. you might get tested on Saturday and then wait. Oh, then you can spend the weekend going through my lecture notes. Then you have a lot of time for that. Okay, so if there are no questions, of course, you can also come up with questions tomorrow morning if something else is coming up. But otherwise, I hope you're dealing okay with the situation. And I will hopefully see you again tomorrow. Have a good afternoon. Goodbye.